Good evening, everyone. My name is Jonny Eintracht. I'm finishing up my PhD with Professor Maria Misigi at UCL Institute of Ophthalmology in London. And we also had a collaboration with Dr. Helen May Simmerer at the University of Mainz in Germany that led to our shared project looking into a potential treatment for Alstrom syndrome. And this potential treatment was geared towards a specific group of patients with certain genetic mutations that we'll talk about over the course of my talk that cause Alstrom syndrome. Unfortunately, Maria is unavailable tonight, so she's unable to attend or speak. But I'm very privileged to be able to be here and to talk to you all about some of the work that we did, and especially to share the screen with Lexi after sharing her inspirational story, and Dr. Paisley for a remarkable lecture. It's very humbling to come speak after both of you. Some of you may remember me from last webinar, and this infographic made by ASCK is a short summary of that. It also shows a little on the agenda for today. So briefly, we'll talk about the cilia and its role in the body, and also in Alstrom syndrome. We'll talk about the genetics of some of the genetics of Alstrom syndrome. I'll give a short introduction to the read-through drugs and their use in our study. I'll go through some of the results from our study, and then also future directions that have come up following our research. So in the beginning, first, I'd like to talk a little bit about the cilia. So this is the part of the cell that changes in Alstrom syndrome or is affected in Alstrom syndrome. The cilia are like the mobile phone signal towers of the cell. And you can see here, if you follow my cursor, this is a picture of the cilia in the lungs, looking a little bit like sea anemones. So their role is to receive and also to send out signals between the cell and its outside environment, and also sometimes between cells. But if the cilia are lost, or if they stop working properly, there are so many important messages that the cells use to communicate to each other and with each other and to the environment that could be lost if the cilia are lost. It's kind of like having a bad Wi-Fi where there's a bad connection. You just can't really, you're a little bit isolated. You can't communicate with anything around you. Cilia can be found in almost every part of your body and take on many different forms. As we were in the Institute of Ophthalmology, we had a particular interest in the eye. So I'll talk a little bit about the role of the cilia, or the cilium in the eye. So photoreceptors are the light-sensing cells in your retina that transmit what you see to your brain, and this helps me to make sense of the world around you and take all the light coming in and make it into a 3D world, which is what we see. And these cells have two major sections. They have the outer segment over here and the inner segment over here. And these are connected by a cilium. So if the cilia do not function properly in the eye, this means that the eye, it cannot send or transmit the signals that it picks up in the light sensing cells to the brain, which means that that you can't really make sense of the world around you, or that you can't construct any images of the world around you because there's an interruption in the signal. So just like the eye, cilia have similar roles in other organs, such as ears, lungs, kidneys, for hearing, proper respiratory function, and kidney function. And if the function of the cilia is lost, it can cause conditions very similar to Alstrom syndrome. So I'm sure it's come up already before, but Alstrom syndrome is caused by a mutation in the ALMS1 gene. And due to this mutation, cells cannot produce a functional ALMS1 or ALMS1 protein that the cilia need to function properly. This means, though, that the cells cannot send or receive the right signals. And we know that Alstrom syndrome is genetic, and it's also a recessive disorder, meaning that you need to inherit both copies of the mutation, one from the mother and one from the father, to be born with the condition. But there is still currently no treatment available, so there is much work still to be done to find a treatment. And just before we go into more of our study, a little slide about what a genetic mutation really is. So in our genetic code, there are 3 billion letters that make up our DNA, and this is divided up into just over 20,000 genes. And if you imagine, if you ask someone to type this all out, to transcribe this, they're bound to make a mistake. So this is what your DNA code would look like in the double helix formation. But if so, just as a human being, if they were typing out over 3 billion letters, that's bound to be some typos. Our cells are no different. And there are some spelling mistakes or genetic mutations found in our DNA. And the Alstrom syndrome occurs if there is a spelling mistake in the ALMS1 gene. And our study focused on a specific genetic mutation known as a stop or nonsense mutation. So cells make proteins from the DNA code, and these proteins, they carry out all the functions that a cell needs to survive and a cell needs to behave to function as normal. And this is done. The DNA is transcribed into mRNA, and that the mRNA is what's used to make the proteins. And a technical term for the building blocks of the mRNA that make the proteins they're called codons, and this is what you can see coming out. You can see on this graphic on the left, each small colored unit, and just like in a traffic light, you have red stop signals and you have green go signals, and the cells also have stop and go signals. But any single change to the DNA code, depending exactly where it is, can introduce a premature stop signal into the code, which means that instead of chugging along like it might here with the codons making the protein, a premature stop signal will mean that the mRNA stops short, the cells stop to make the protein that is needed, and then eventually the protein is lost because the complete code required to make it is lost from the cell. But there are certain drugs that can almost hack this system if there's a premature stop signal in the cell. So just like this roller skater through New York City that's hacking the traffic lights, changing the red to green, there are some drugs that can do the similar thing. And what they do is that they tell all the cell machinery to still make the protein, even if there's this premature stop signal introduced due to a genetic mutation. And this means that cells can still produce large amounts of the protein required despite the mutation, and still function as they should. Roughly 40% of ALMS1 mutations are these stop mutations where a premature stop signal is introduced into the DNA code. And this means that roughly 40% of patients may have little to no ALMS1 or ALMS1 protein expression in their cilia. So just to illustrate this point, this picture of a cell, this picture on the right is a picture of a cell. So the blue is the nucleus, and to the right is the cilium. So the cilium is the green shape here, the green string that you can see here, and at the bottom is the ALMS1 protein in red. It's this red dot, and this is kind of like the base. This is at the base of the cilia of the cilium, and it's kind of like the base of a phone tower connecting it to a grid. So it's essential for it to maintain its connection and to be able to interact with other cells in its environment. However, patients that have, as I mentioned before, patients that have this premature stop signal may have little to no LMS1 protein, so they lose this red dot in their cells. But the potential of certain drugs means that we have a chance of being able to treat these patients and helping them remake the ALMS1 protein and allow the cilia to function again. So we looked at two specific read-through drugs, the drugs that can read through a premature stop signal. The first one is known as Ataluren, or PTC124. And it can read through the stop signals or like kind of hack the traffic light system. And this drug has already been approved to treat Duchenne muscular dystrophy that's caused by similar stop mutations like Alstrom syndrome. And importantly, it's proven to be safe and effective, and it can be taken orally 3 times a day. So it also comes in a powdered form, so you can just mix it in water or juice or milk or something and drink it and take it like that. Amlexanox has a slightly different history. In the past, it's been used to treat ulcers and asthma, but following a very intensive study of many different drugs, a group of researchers, I think in France, found that it had the similar ability as Ataluren to read through the stop signals in the DNA, and to tell cells to still make proteins, even though a premature stop signal to tell the cells to stop making the protein was found. But to test out the effects of these drugs on the ALMS1 gene and restoring ALMS1 expression despite a genetic mutation, we needed to get some patient cells in the lab. So to do this, we had some very kind patients donate their skin, or donate a small punch of a small bit of skin from their upper arm, taken by a little biopsy punch, and we placed this punch of skin. It measures maybe 5 millimeters in length, so it's quite small. And we placed it in a dish full of nutrients and specific conditions to help the cells grow in the lab. And they grew over a few days into something similar to what you see on the right of the screen. This is an image of Alstrom syndrome skin cells from a donor patient. And these are cells that we grew in our lab. And they were fed with a specific mix to keep them happy of nutrients and other molecules. And into this mix of nutrients and molecules, it kind of looks like what you can see in the bottom right image, almost like a red juice. And into this juice, we can put the drugs also in the cells, as just like they're taking the nutrients that are in this mix, they also take up the drugs. So we knew that we had a way to get the drugs into the cells. And our research, our study focused on 4 main questions. So if we're going to talk a little bit about our project now. So we first wanted to see whether we could actually detect the ALMS1 protein at all. This was in patients, or in healthy donors that didn't have an ALMS1 mutation. And if we could detect the ALMS1 protein in healthy cilia, we could also show that if there is a mutation, the ALMS1 expression is lost. And this is also very important to see whether we could restore the ALMS1 protein following treatment of patient cells with the drugs. And this was the first objective or output that we aimed to find. We then wanted to see whether the cilia looked or acted differently in the patient cells compared to those without the mutation. And if they did, could treating the cells from the Alstrom patients with both drugs, the Ataluren and the Amlexanox, help them function properly again. So to answer the first question, we found that we could in fact detect the ALMS1 protein in healthy donor cells in our lab. And again, it looks similar to what I showed before, as a red dot at the base of a nice looking green cilium here. But you can see that in cells taken from a patient, from an ALS1 patient with a stop mutation, introducing an early stop signal and the protein not produced. In fact, we don't see this red dot at all in the cilium, suggesting that the protein is lost because of the mutation. But remarkably, we found that once we treated these patient cells with both drugs, both ataluren, PTC124, and amlexanox, this little red dot, the ALS1 protein, returned to the base of the cilium. And this happened in roughly 40 to 50% of cells. We then had another look. We looked to answer question number 2, like, did the cilia look any different, or did they appear differently? So we looked at as many cilia as we could find in our cell samples, and we measured their lengths, and also we counted the amount of cells that actually had cilia. But it seems that the length of the cilia between all the experimental groups, and also the amount of cells that actually had a cilium on, that didn't really change or differ between the cells without the mutation or the cells with the mutation. And this suggested to us that perhaps the ALS1 protein doesn't really affect how the cilia are made, but perhaps it affects how they function. And we found that the answer to that question is, in fact, yes. We looked at a specific transporter protein known as IFT88, and that's involved in transporting the signals up and down the cilium. And this is this nice green dot. You can see in the top panel where the white asterisk is. And there's a large dot here and a small dot just to the right. And you can see that it sits nicely back, but again, at the base of the cilium and then also along the cilium, showing that it works both at the base and in transport, taking things up and down. But again, we found that in patients, in cells that contained the ALMS1 mutation from patients, this transporter protein IFT88 was lost. And just like you couldn't see the ALS1 protein in the slide before, you can't see this IFT88 protein connected to the cilium in the ALS1 patient cells here. But again, we found that after treatment with both ataluren or PTC124 and amlexanox, we could restore expression of this transporter protein back to the cilium. And this suggested that treatment with these drugs can potentially also help cilium recover their function and not just recover expression or restore expression of the ALS1 protein. We looked at another protein to assess changes to how the cilia function due to the mutation and if this can be restored after treatment. And this protein was called SSTR3. And this protein is actually, this is the actual cargo that's transported up and down. So previously we looked at the machinery that transports cargo or signals, and now we looked at something that's actually transported up and down the cilium. And we found that in our wild type healthy donor cells that do not have the mutation, this protein cargo SSTR3 was found at the base of the cilium, with a similar pattern to how the ALMS1 protein is expressed. But we saw that in cells with the ALMS1 mutation, this SSTR3 protein was just slightly to the left of the cilium. It's moved slightly away from the base. And this is almost like an analogy that I thought to explain this phenomenon was like having lost luggage on a flight. So your luggage may get on a plane to an exotic destination, but a very different destination to where you wanted it to go. But we found that once we treated these ALMS1 patient cells with both drugs, again, we could restore the SSTR3 expression to the base of the cilium, putting the cargo back to where it needs to be, and helping the cilium send the right signals to each other and also to the environment. And this is almost like having lost luggage reunited with its owner once again. So to sum up some of the results that I've been talking about, we currently have there is no existing treatment for Alstrom syndrome. And any stop mutations in the LMS1 gene that introduce this early stop signal cause the cells to stop producing the LMS1 protein, as we saw that it was lost in the cilium. And they also affect how the cilium functions. But what we saw from our study was that treatment of patient cells that with the mutation with two read-through drugs, Athlone and Alexidox, they remarkably rescued the expression of the LMS1. So we found the LMS1 protein again in treated cilium. And they also restored correct function to the cilia again, allowing them suggesting that the transport machinery and also the signals that are transported by this machinery can return to the cilium. And this is kind of like good Wi-Fi signal returning after being lost. So it started off good, then it was lost, and then after treatment, this good Wi-Fi signal comes back again. We were very fortunate actually to be able to publish this study in the journal eBiomedicine, part of the Lancet group, one of the more prestigious medical journals in the world. And we can send out links to the study if anyone wants to read more. We're also happy to talk more about the study. But if you can search. If you do a Google search of my name or the lab or just my name in LMS1, you'll be able to find this study, but also through ASCK, you can get access to this paper. So then you could also say these results are great, but then there's a lot more work to do. Like, what comes next? So we have a few ideas that could be helpful to expand and also advance the study. So one of the things that I think would be helpful would be to expand the study to include more patients and different stop mutations, as we only worked on specific mutations and only with a limited number of patients. And we also think that it would be important to work on other tissue of the body that are affected by Alstrom syndrome, as this study only focused on skin cells as more of a proof of principle. Importantly, also, for any chance that drugs can make it as official treatments on the NHS or similar governmental or public healthcare systems, they would need to go through clinical trials beginning with phase 1 and phase 2. So Maria, my supervisor, has currently been given some Athlone to trial on choroideremia, which is an eye disease, and also on Usher syndrome. And Usher syndrome specifically has some similar features to Alstrom syndrome and is also caused by these stop mutations, where the Usher syndrome patients don't produce the right protein that they need. And if we see that in these trials, oral delivery of the drugs can help Usher syndrome patients and restore any changes to the function of their cilium, it's a great step in the right direction for getting these treatments for any future trials with Alstrom syndrome patients, as we can show that these drugs indeed have an effect on patients that have a similar condition in their cells. Another thing that I guess the final direction that we thought to go was to expand the study to more complex models in the lab. As I mentioned before, we only work on skin cells, but our lab specializes in creating retinas in a dish, which is eye tissue very similar to like the developing retina during pregnancy. So what we thought to show was that perhaps these models can act as a platform to test these drugs to see whether the light sensing cells that are lost in Alstrom syndrome can then function again in the retina. And these these images are from a paper that we published from our lab showing our own lab-grown retina. And these images are taken at earlier time points, such as this one on the left is at day 35 of growth, and this is the one on the right is at day 50 of growth. But in fact, the retinas can be growing for over a year. And as they grow, more mature and complex photoreceptors, which are the light-sensing cells that degenerate in AS, grow. And we think that these can perhaps act as a relevant model for more sophisticated studies into Alstrom syndrome and allowing us to expand our current work. For any other information on Alstrom syndrome, we also have a website called Gene Vision. And this has plenty of information about Alstrom syndrome and other conditions. So if you want to know a little bit more about it or other genetic eye conditions, please have a look at www.gene.vision. As we get to the end, it's very important, maybe this is the most important slide, just to say thank you to a lot of the brilliant scientists that we've been fortunate to work with, or that I've been fortunate to work with, that have made this study possible. I'd like to say thanks to my team at UCL, my supervisor, Professor Maria Musaji, and everyone there that made it possible. A really big thank you to Dr. Helen May Simra, who was our main collaborator. And without her, this project would not have got off the ground or been as successful as it was. Another thank you to the team at UCL Great Ormond Street at the Institute of Child Health, particularly Dr. Elizabeth Forsyth. And I also wanted to thank Dr. Rob Semple and Dr. Jinhua Chen for their expertise and also for the patient cells that they provided to us. Again, without them, this study would not be possible. Also, wanted to say thank you, acknowledge our funders for the financial backing for this work, and most importantly to the patients and their families for going on this journey with us. And none of this work would be possible without your input and your interest and your investment. So really, really thank you from the bottom of our heart. And I just want to end saying that if as any Alstrom syndrome patients that want to be in touch or want to be more involved in the research side, so please reach out to either myself or Maria. Our email addresses are here, and we can send them out after the webinar. I've just put some pictures of other patients that have come to donate some skin at Maria's clinic at Moorfields. And we'd love to have more Alstrom syndrome patients come in and participate in our research, really at whatever capacity feels comfortable. And this can be either to donate cells that can then be grown up and tested in the lab to be part of similar studies that I presented tonight. But there's also a need for patients just for natural history studies, and this can help us track the progression of Alstrom syndrome. And then, importantly, to be able to create objectives for clinical trials and to show what improvements drugs can make or what outcomes we can aim for. And you can also talk to anyone at ASUK to know more, or to find out further information about research participation. But again, I wanted to say thank you very much for your attention. And again, it's a real privilege to be able to present here tonight with such inspiring people. Thank you very much.
Perfect. Thank you very much, Jonny. And it's always fabulous to see some of the start of some of the development work. So huge thank you for sharing that with us tonight. I'm going to go to Laura and try and describe that graphic a little bit. We have got some questions coming up for you and some for Lexi. But I did just want to reiterate my thanks to you, Jonny, for, I know you don't know this, but for working for Alstrom. No, you don't know you're working for us, but you certainly are. So thank you for that. Okay. Can we share that screen, Laura, full size, and I will try to describe it?
Yeah, working on just trying to find it on my desktop.
Okay, whilst you're doing that, I've got a question from Kerry to Jonny.
Yeah, great presentation. I just, because obviously we'll be looking to use these as repurposed drugs, so they've clearly already got that safety profile. I just wondered why you were looking to trial them in Usher and choroideremia. Is that more around the age of the patients? Are you looking, will they be adult patients or children, young people?
Those would be adult patients. And I think also more specifically for our group, Maria has a bigger history in working with those conditions. And also she has a larger patient bank at Moorfields. So there's a lot of logistics in like pitching for the trials and being able to get companies on board. So that's really like one of her areas of expertise. And like our I guess our foray into Alstrom is much newer, and we have less experience in that. So if we can show, like, if we have like a good proof of principle of good clinical outcomes in patients that Maria has a lot of experience with, then we'd look to say we want to expand into other work that we've started to do.
Yeah, okay. And just to let you know that I did speak to our ophthalmologist after you presented at the global conference in December, and they are keen to support kind of data collection, really trying to build up that natural history. So maybe we can get together and have a chat afterwards and talk next.
Okay, fantastic.
Perfect, thank you. I have another question for you, Jonny, which is in the chat, which is one of our folks in the States, and she says, is there a way to participate in the research being in the US?
I mean, the capacity for patient participation, for sure, it exists worldwide. So if there are labs that are doing this kind of research in the US, it's also so that I would recommend to reach out. I mean, it's also possible to reach out to us, and if there's a way, if there's someone in the US that can take a skin biopsy or some sort of cell sample, it's also possible to freeze down those cells and ship them over to the UK. But I'm sure I can also look if there's any groups in the US that are doing similar work, and if I find anything, I'll pass it on.
Okay, thank you. And there's a fairly can you see any of these questions where you are, Jonny?
Yeah, I can see on the chat here.
Okay, can you see the question from Chase Palmer?
Yes. Does it seem likely that I know it's not really relevant to improve function in vivo?
Want to kind of read it out loud so that we can.
So does it seem likely that amlexanox could be rebranded to improve ciliary function in AS in vivo? So more in animal models. Amlexanox paste was discontinued in the US, but the crossover between the two indications does seem relevant. Also, is the amlexanox, I guess, a mechanism of action known for the improved ciliary function in vitro?
Okay, so Chase is a pharmacist and works and heads up ASI in America.
Oh wow, that's a great question. Both are great questions. And in vivo studies, it's definitely been on our mind as one of the next steps. Again, for any significant data to go into, for like pushing for any patient trials, you need good in vivo data. One of the things with amlexanox is we'd have to work out like how to dose animals so that it can reach like systemic dosing so that the drug can reach all affected tissue in the animals. And we don't know whether that again would be old administration like atoleurin or with injections. But we'd have to do some preliminary tests to see if that's possible. The mechanism of action for improved ciliary function, I think my guess would be that because it's able to read through the stop mutation in the ALMS1 gene, restoring the gene seems to be able to restore some of the function. The interaction between the ALMS1 gene and some of the genes expressed in the cilia are less known, and that's also a fascinating, I guess, puzzle to be able to try to put together, just to see how all these genes work together to create a functional cilia.
Okay. Thank you. We are going to capture a lot of the information tonight, and we will try and write it up in quite simple terms, as simple as possible, which kind of helps all of us. So thank you for that, Jonny. Thank you for the question, Chase. Laura, can you share your screen now? Okay. So can I get you to... Okay, thank you. So, so on screen at the moment, we've got a little graphic capturing Jonny's talk, which talks about ALS1 is a genetic mutation inherited from both parents, and just some of the discussion around what his presentation was around. But we've also included on that the website www.gene.vision, which some of you will be, you'll be able to access that from hopefully anywhere in the world. So thank you very much for that. And thank you, Jonny, again, for that very interesting and very hopeful presentation. Can I now, Laura, to go and give me the graphic for Lexi? And I've got a question for Lexi. Hope, if I can. Thank you. Okay.
This one is very much work in progress. I wanted to get her words in, and there will be a lot more illustration.
Okay. Thank you. So I've had a question, Lex. Can you hear me okay?
Yes, thank you.
Okay. So this is from a dad, and he says, Lexi, are you able to share some more information about your educational journey? This is something that parents struggle to navigate, but it would be wonderful to hear from the horse's mouth. He says, pardon the pun, what your educational challenges and successes have been. He has a son, Teddy, who's 8, who has some sight, some learning difficulties and mild ASD. And he says, congratulations on your great news. I wonder if you just want to give us a kind of bit of an overview of your education journey.
Sure. So in the UK, I went to a mainstream primary school, which had a special educational unit within to give me support as a blind student. But I had sort of, a one-to-one support in class. I then went on to secondary education in a special school just for visually impaired students for 4 years, but then came back to mainstream for my higher education, so my GCSEs and A levels. I think education is challenging. Technology definitely plays a great part, I think, in education when you have special needs. It's difficult to access sometimes the technology that you need, and can be a real fight. But I think, in this day and age of sort of iPads and laptops are very much a common aspect in schools. There's a lot of technology that can help visually impaired and hard of hearing children to gain education and access to the information. Education was tricky. I tried to do higher education as well and found this extremely difficult. But I also know that we have a lot of adults who very successfully access higher education. So I think it's a real mixed bag. I think for me as a child, I was very lucky that I had parents who pushed and fought a lot to get me the things that I needed. And that's where AS UK can help, as well as the other Alstrom societies around the world to help support the parents in that fight for the rights that their children have to education and to access their education. But I also had things like design for living skills, so people that helped me to learn things like cooking, cleaning, those sort of more activities that aren't necessarily academically educational, but were very much a help to me and to making me independent. Things like mobility. I also had as well throughout the time and places like guide dogs. They do cane training as well as the dogs. So again, they helped with mobility and educations, and establishments like that can go into schools and ensure that your child is getting the right help and support from all those sorts of different places as well.
Lovely. Okay. Thank you, Lexi. And maybe, just thinking through, maybe that's for one of our kind of bite-sized webinars that we do, perhaps something around education might be of interest to parents. So let's hold on to that one and see if we can come back to it. Okay, a couple of more questions for you, Jonny. One of our ex-chair and ex-trustee of AS UK says, you'd be happy to talk more about participating in research, and would you be able to put some information about how we could possibly participate and share that with AS UK to be passed on, which I'm sure we can coordinate. And again, from from Palmer in Canada, talks about all information regarding the eye would be excellent because he's in great need of an eye doctor. And Jane asks, does the cilia or protein cause fibrosis in organs?
I'm not sure of the answer to Jane's question. That I'd have to check out.
Okay.
I can follow up with her.
That's fine. I mean, we could, as I said at the beginning, there's always questions we'll have that perhaps we may not immediately know the answers, but we can follow up as well and find out and follow that through and give Jane an answer when we send out the package of information after this. Okay.
Sounds good.
For that, let me just see if there's any more questions. Great. Palmer's come in and said happy to discuss his education experience as well. So brilliant. Thank you for that, Palmer. Let's hold on to that one for another webinar. And he says he wants to share the information with his eye doctor. And that's really helpful because these webinars and certainly these mini learning modules that we do, please do share and the graphics with those who may be involved in the support of you as a person with Alstrom or a family member. Please do share them because that's part of our awareness raising, learning and development of this ultra rare disease. I'm just going to check with Kerry if there's any more questions that are on there that I've missed, perhaps.
No, I think there's just things on there that we're definitely going to follow up with. We will send that out next week. There's links to different research papers and things that people have asked for. We'll save all of these question and answers and queries and things, and we'll do lots of follow up. But, if there is anything that you think of after the webinar that you want to ask, just send us an email and we'll get your questions answered by different members of the team.
Okay, and that email address is generally to Catherine. You'll see that on the information we send through. She's just not here today and tomorrow, but we do get back to you. Okay, so it falls to me to just kind of round up and say a very big thank you to Dr. Richard Paisley. That was a fascinating insight into the history, and I'm always fascinated, Richard, to listen to you because you have this extraordinary experience and knowledge and funny stories. So huge thank you to you.
Thank you.
And a great way for us to celebrate 25 years of AS UK. It is very much from little acorns that we've now got this really, we still call ourselves a small charity, but actually if you've seen the kind of photographs of the trustees, of the staff, of the volunteers, we're growing and growing, which is brilliant. So thanks everybody for their support with that. I was particularly interested, Richard, when you talked about Ethel Burnside collecting all this information. So another shout out really for this patient registry that has been developed, because something that is so important is collecting that data. And I know it's a bit kind of, it can seem boring and it can seem something in the future, but it's so valuable to clinicians and researchers. So well done, Ethel Burnside. Good for you. A big thank you to Lexi. And yeah, what can I say about your news? Well, nothing really, except I'm so excited.
Thank you.
Thank you to Jonny for sharing that and for working with us in terms of looking at, you know, early development, but fabulous research nonetheless. If you've enjoyed tonight, tell others about it. Tell your folk who are educating your children, any social workers, colleges. Please do share those mini modules around diabetes and all the other things that we've done on webinars. And if you have any ideas for a webinar, you want us to focus on something such as education, send it in to Catherine. We can fairly quickly put something together and get somebody who can talk about their experience, but sometimes with an educator too. So please do that. We do share the graphics and we encourage you to share them too, either on Facebook or if you're on LinkedIn, because the more people that know about this ultra rare condition, the more we can learn, the more knowledge we get. We are delighted to tell you that we are doing a webinar at the end of this month in Spanish. So for Spanish speakers, you're very welcome to join us. We're just hosting it for the Spanish Association of Alstrom Syndrome. So very pleased to be helping them do that. The USA conference I've mentioned is October 2023. If you want more information, ASI will have it again. Should you not be able to find their details, come to us and we'll connect you together. I mean, that's kind of very much part of what we are trying to do. We will follow up with this afterwards, and we will look at trying to, this year, we're going to do a couple of webinars on the Alstrom ear and hearing, and potentially if Professor Rick Steeds is still online, something around the Alstrom heart. So we've things that we are already lining up, but really want to hear from you in terms of exactly what you want to do. Okay, so I now ran through that really quickly. We put these webinars together afterwards. We've put a few top tips together so you can revisit, or you can send them to friends and family and others that you think would benefit from having, you know, a kind of bite-sized learning module around subjects, and particularly perhaps the history of Alstrom syndrome. So that's our past, present, future. Now, if I don't have, unless I have any other questions, let me just check.
Just going to finish with one comment, Anne, from Jane, John James's mum. She's put, thank you so much. 23 years down the line as a parent, and I've learned something new.
Fantastic. Okay. Kerry, would you like to finish off as the Chief Exec of AS UK?
Just thank you everyone for joining. I think it's, you know, thanks to you, Anne. I think it's great to see the global community coming together. And I think just to echo Richard's theme running through his presentation, collaboration is key, and we're very keen as an organisation to collaborate with people, you know, within the Alstrom community and beyond, because you know, we're too rare to work in isolation. It's only by working together, I think, that we're going to move things forward. So thank you to everyone, and look forward.
Perfect. Lovely. Thank you. Thank you very much, Jonny. Thank you, Richard. Thank you. See you next time.